Hello everyone, and thank you for attending today's webinar, Companion Diagnostics Biomarker Webinar, Importance, Trend, and Forecast. I'm Vivian Xie, Digital Editor at Bioanalysis Zone, and I'll be your host for today's event. Before I introduce you to our speakers, I'd like to quickly cover a few housekeeping items. All the widgets are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right-hand corner. Some networks cause slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help widget at the bottom of your screen. An on-demand version of today's webinar will be available approximately 1 hour after the webinar and can be accessed using the same audience link that was sent to you earlier. We are delighted to have Kai Wong from Frontage Laboratories presenting for us today. Dr. Kai Wong is Director of Bioanalytical and Biologic Services at Frontage Laboratories and is leading the biomarker services of Frontage. Dr. Wong earned his PhD in chemistry from Rutgers University, New Brunswick, and received a Bachelor's of Science degree in chemistry from Nanjing University. Thank you again for joining us today, everyone. I'll now hand over to Kai Wong to start today's presentation.
Hello, everyone. My name is Kai Wong. I'm Director at the Biomarker Services at Frontage Laboratories. So thanks for attending today's webinar. I'm going to talk about the companion diagnostic biomarkers. So here's a brief outline of today's topic. So I'm going to do a quick introduction of precision medicine and companion diagnostic. I'll also talk about the success of companion diagnostic in recent years. And more importantly, I'll do a introduction on the companion diagnostic in breast cancer, lung cancer, and Alzheimer's disease. So last but not least, I'll talk about the precision medicine and companion diagnostic outsourcing strategy and forecast. So precision medicine, also known as the personalized medicine, is a medical model of customization of healthcare, including diagnostics, medical decision, and treatments which are being tailored to a group of patients. So precision medicine actually originated in oncology in the 1990s. It has made significant progress in the past few decades in other disease categories such as the neurodegenerative disease, cardiovascular disease, and even psychiatric disease. So on the other hand, companion diagnostic has very close ties to precision medicine. They go by hands. So according to the FDA, the companion diagnostic is a medical device, often an in vitro device, which provides information that is essential for the safe and effective use of a corresponding drug or biological product. The test actually helps a healthcare professional to determine whether a particular therapeutic product is beneficial to patient and will outweigh any potential serious side effects or risks. So precision medicine actually made a significant progress in the past few decades. So according to the US FDA, the ratio of approved personalized medicine over the total of approval new drug entities since 2005, it increased from 5% to almost 40% in 2020. So in 2020, actually, there's 19 new therapeutic entity that was approved, and that is almost 39% toward the overall approved new drug entity, which is 49 in total. Moreover, from 2017 to 2020, precision medicine approval achieved over 30% of the overall approval rate by FDA in 3 out of the 4 years. Also, there's a total of 45 companion diagnostics in associate with the precision medicine up to today. And as for the platforms, the PCR is the most used platform. That is the 16 of the approved companion diagnostic use the PCR platform. followed by the IHC, and which is 12, and ISH. There's a 9 platforms. So NGS is a fast-growing platform in recent years, and there's also one imaging technique tool for the companion diagnostics. So precision medicine actually relies on various types of technology for the identification and quantitation of patient biological information, including the genome and protein. Eventually, it leads to the diagnosis and treatment of disease. So, as stated in the previous slides, the PCR is one of the most important tools to study the genome sequencing and RNA sequencing for the potential patients. You can actually get the DNA or RNA mutation information out of that. So eventually it leads to the disease identification, diagnosis, and treatments. On the other hand, in the recent few years, because of the development of the technology, so more and more immunoassays are being used as diagnosis tools. So the immunoassay actually can be used to detect abnormal protein biomarker level, and this, which is also potentially associated with the particular disease and subsequently the treatment. So up to June 2021, there is a total of 45 companion diagnostic assays that has been approved by the US FDA. So out of the 45 assays, significant amount of these assays actually are for cancer treatment. So essentially there's 42 out of the 45 are for cancer. And for these cancer treatments, lung cancer and breast cancer takes up a majority of these assays. So there's 12 approved diagnostic assays for lung cancer, and there's 17 for breast cancer. So the first companion diagnostic test was developed in 1998. It is actually for a drug called Herceptin. In the same year, the Herceptin was approved, and this companion diagnosis was used to detect the protein HER2, and it's essentially used to detect the abnormally high HER2 protein or gene level in breast cancer patients, and also detect the response of Currently, there are 17 FDA-approved companion diagnostic tests for breast cancer, majority of which targets the HER2 protein. So these tests, again, the protein HER2 test was established as companion diagnostic to identify the abnormally high HER2 protein or gene level in breast cancer patients, and the treatment response with Herceptin. So the precision medicine for breast cancer, however, is still in a very early stage, and there's many challenges down the road. So, for example, the triple-negative breast cancer, which is one of the most malicious type of breast cancer, it still lacks the true precision medicine due to various type of reasons, including the heterogeneity of the disease itself. So currently, the precision medicine only targets about 20 to 30% of the overall breast cancer cases. The second disease I want to talk about is for lung cancer. So pre-COVID, lung cancer was the second most common cancer in both men and women. For men, it is second to the prostate cancer, and for women, it's second to breast cancer. However, as for the fatality, each year, more people die of lung cancer than the colon cancer, breast cancer, and prostate cancer combined. For lung cancer, the early diagnosis is extremely important. And in order to achieve that, a biomarker screen is actually able to help. So lung cancers, especially for the non-small cell lung cancers, it's actually highly influenced by genetic differences between patients, therefore causing the differences in the metabolism biomarkers. Identifying the personalized biomarker will help to create the maps for healthcare professionals, doctors, to personalize the treatment and medicine. Here I want to talk about one technique is called the liquid biopsy, aka liquid fluid biopsy. It is the analysis of cancer biomarkers in biologic fluids. It was first introduced in the early 2000s and this becomes a very important tool for cancer diagnosis. Also, I want to talk about the biomarker itself. So for certain type of patients or disease, not only one biomarker is sufficient. Normally for healthcare professional or doctors, they look at a biomarker panel, including a few types of biomarkers to increase the selectivity, sensitivity, and accuracy for the diagnosis and treatment. So again, for lung cancer, here I want to show you the biomarker panel. There's several types of biomarkers for lung cancer, including the ctDNAs, the metabolites, typically small molecules. There's also genomic biomarkers and protein biomarkers. Each type of these biomarkers actually have their own challenges for the detection. So for example, the ctDNA is extremely low in concentration in human body. So in order to detect these ctDNAs, you need a very sensitive instrumentation. And also ctDNAs experience the challenges that it has actually very high variability among different people. So metabolites as a small molecule, sometimes there's a stability problem, sensitivity problem. For genomic biomarkers, there's also sensitivity requirements. For protein biomarkers, usually it requires special assay kits. There's actually a fairly good amount of good assay kits from different type of technologies. However, for this type of immunoassays, the selectivity and the sensitivities are always an issue. This is an example of the lung cancer biomarker panel. So again, there's in order to detect the lung cancer and make the correct diagnosis, people look at a series of biomarkers to constitute a whole biomarker panel. For this lung cancer, there's different types as shown in this figure. Typically, there's EGFR, KRAS, these genetic changes, and also for these type of genetic changes, there's a companion diagnosis actually approved by US FDA. So there's also other panel biomarkers such as metabolic biomarkers, including the amino acids, fatty acids. There's also several types of proteins, the antigen, antibodies, etc. So with all these combined, you actually have a very increased sensitivity and specificity for the whole panel. As of today, there's 12 companion diagnosis assays has been approved by US FDA for non-small cell lung cancer. So these tests look for genetic mutations such as the EGFR, KRAS, or protein biomarkers such as PD-L1. Potentially, blood-based biomarkers such as the ctDNA can also be non-invasive diagnostic tool or of becoming companion diagnosis. For the research, next generation sequencing, the NGS, is the most useful and powerful tool to exemplify the non-small cell lung cancer as a perfect example for the precision medicine. Small cell lung cancer actually also started to begin to enter the precision medicine year recently, and there's some publication coming out in 2020 and 2021. So as you can see, the biomarkers are extremely important, not only because it just be used as companion diagnosis, but also for other reasons. So biomarkers actually help disease diagnosis and prevention. It could be used as a routine diagnosis test. if FDA approved as in vitro tests, IVD, or laboratory-developed tests, LDTs. So biomarker provide the potential targets for new therapies, indicate and monitor the drug efficacy and safety in drug development, monitor the patient response toward the drug. So based on the purpose, biomarker can be characterized as diagnostic biomarker, predictive biomarker, safety biomarker, PD biomarker, prognostic biomarkers. So biomarker actually improved the drug development success by over 200%, as you can see in the chart in these slides. So according to US FDA, the major challenge nowadays for pharmaceutical development is the failure rate. With the help of biomarker, as you can see in this figure, at different phases of the clinical trial, with the help of biomarker, it significantly increased the success rate. So overall, from phase 1 to approval, it increased the success rate from 8.4% to 25.9%, which is over 200% of increase. So that significantly saved a lot of money for the pharmaceutical company, and also helped the patient to get the drugs much sooner than before. In bioanalytical lab, we typically deal with complex biomarkers, majority of which are soluble biomarkers. So the biomarker itself, it could be metal elements such as copper, iron. It also have small molecule biomarker types, proteins, DNA segments, cells. They all could be biomarker targets. As for the matrices, biomarker can be identified and quantitated in blood samples, urine samples, CSF samples, saliva samples, cell culture, tissue samples as well. For our CRO, when we set up a biomarker assay for diagnosis purposes, we do full method validation under the guidance of GLP, and then we also follow the requirements for CLIA. So with both GLP and CLIA, you got a biomarker full validation for the diagnosis purposes. So biomarker actually is associated with various types of disease, including the oncology disease, infectious disease, cardiovascular disease, endocrine, neurosciences, inflammation, respiratory diseases. As mentioned earlier, so biomarker should be looked at as a panel, and including the genomic markers, protein markers, metabolic markers, as well as analysis of ctDNAs. So with a combination of different types of very selective biomarkers, it actually could provide an optimal panel of information for the healthcare professionals to effectively diagnose and treat the patients. So the third disease I want to talk about is for Alzheimer's disease. So Alzheimer's disease, based on the pathological process, it could be classified as A, N, and T. So A is for amyloid pathological process, T is for tau protein pathological process, for N is in general for neuron degeneration. So for each type of these pathological process, there's associated biomarkers. For example, for amyloid, there's A beta 38, 40, 42. For the tau proteins, there's total tau, phospho-tau, including some of the subspecies for tau, phospho-tau 181, phospho-tau 217, 231. For neuron degeneration, there's even more different types. For example, for inflammation, there's TREM2, YKL40, GFAP. For neuronal injury, there's a neurofilament light chain, NFL. There's a VILIP1, FABP3. There's also several different types, such as neuron granin, SNAP25, and PTX2. So based on the different types of biomarkers, here I want to give you a full overview of the biomarkers associated with Alzheimer's disease. There's genomic biomarkers, there's also protein biomarkers, metabolic biomarkers, and others. So especially I want to talk about for the protein biomarkers, because of the low concentration of such protein biomarkers. Traditionally, there's a very high difficulty in detecting these markers. However, with the development of the instrumentation technology, there's several types of assays that's being set up on ultra-high sensitivity instruments, such as the quinteric Simoa, especially for the neurofilament light chain, phospho-tau 181. These are being detected and quantitated using these quinteric Simoa instruments, and it's actually revealed a lot of information for Alzheimer's disease diagnosis as well. So here I want to show you a popular neuron degenerative protein biomarker assays. So as stated in the previous slides, there's total tau protein in human CSF assay. This can be detected in quinteric Simoa. Neuron filament light chain can be quantitated in human plasma CSF serum. It can also be quantitated using quinteric Simoa. Phospho-tau 181, which is very new assay and popular assay, which is also Simoa assay. The phospho-tau 217 has been reported by multiple literatures. For this assay, there's one monoclonal antibody was used for all the publication, but we're also developing this specific assay because of its high demanding and very useful assay for Alzheimer's disease. There's also NFL, GFAP assay on Simoa, YKL40 as ELISA assay. All these type of assay are very useful for the Alzheimer's disease because of its high sensitivity and selectivity. So precision medicine and the companion diagnosis, again, for Alzheimer's disease, there's currently still no clinical proven effective treatment for it. So this statement was true until yesterday because US FDA actually approved the first Alzheimer's disease drug from Biogen. This is the first approved drug in the past few decades. Because of the rare treatments for Alzheimer's disease, there's currently no companion diagnostic test available. So again, there's a lot of research scientists working on this day and night to actively identify the potential biomarkers, especially for the neuron degenerative biomarker, to benchmark the proposed pathological mechanism. And these markers are actually potentially very useful candidates for the companion diagnostic tests in the near future. So as for the future development for the companion diagnosis and the precision medicine for Alzheimer's disease, there could be various human genes that contribute to the pathogenesis, which will require, again, extensive research process and efforts to identify and locate. So last but not least, I want to talk about the precision medicine and companion diagnosis outsourcing strategy. So there's actually two different types. The first one is a three-way partnership, including the pharmaceutical company, the in vitro test developer, and the CROs. So in this strategy, pharmaceutical companies actually outsource the tests into a CRO, but the CRO will also sort help from an IVT developer to provide the FDA-approved in vitro tests. So in this type of partnership between each pair, there should be open communication. There should be collaboration in order to make this very smoothly. There's the second one is a two-way partnership, including only the pharmaceutical company and the CRO. And as a CRO, they will set up the diagnostics test at their own and sort for the approval from the US FDA, and it will be used for at the multiple sites depending on the location of the clinical trial. As for the outsourcing, there's a lot of things to consider, including here I want to talk about is the experience and expertise of the partners. So some of the lab are very experienced on such type of diagnosis tests. The others are not so much. So the pharmaceutical company actually should really be careful in choosing the partnership, which could significantly expedite their whole process if they choose the right partners. There's also regulatory compliance experience needs to be considered. Geographic coverage is also very important. For example, if this pharmaceutical company is sorting to outsource its test for a global clinical trial, then this partner of CRO should be capable of doing the test on different locations all over the world. There's also cost control needs to be considered. Commercialization as a final stage of this type of tests is also very important, and the timing of the partnership is also very important as well. So as a CRO, Frontage actually have a very broad capabilities in support of the biomarker immunoassays. Here I just want to give you a brief outline of the techniques of the instruments that we are currently using in support of various type of diagnosis tests. So that's including the SpectroMax for ELISA assay. There's also, I just wanted to talk about a few key instruments, including the MSD S600 imager. There's also Quanterix Simoa HDX analyzer, ELA proteins simple system, Quantis QView imager. There's also Luminex instrument, Gyros instrument. All these type of instrument is actually can be used to detect the protein biomarker concentrations. For qPCR, ddPCR, NGS instrument, it's for the genomics information discovery tests. So this shows our capability and expertise in the biomarker analysis. So in summary, the precision medicine and companion diagnosis is undergoing exploding development in nowadays. So according to the estimation that precision medicine market size is going to grow from 58.5 billion to 98.5 billion from 2020 to 2026. So as you can see, the market size almost doubled up in the next few years. From scientific perspective, the NGS will keep playing major roles to uncover genomic information for potential companion diagnostic tests. Immunoassay will play more and more important roles as diagnostic tools for protein biomarker using various type of technologies. So precision medicine and companion diagnostic will dive into more and more disease categories in addition to the oncology, and there's still a lot of challenges for pharmaceutical companies and the CROs, including the demand for flexibility and agility, urgent needs for more and more talents because of the new technologies keep showing up, and standardization and regulatory compliance harmonization, often the new technology, etc. Thank you very much, and that's all for today's presentation.
Thank you, Kai, for that very insightful presentation. I hope our audience found it of great interest and value. If you do have any questions for our presenter, please submit them to webinars@bioanalysisZone.com. I would like to once again thank Kai Wang, as well as you, our listeners, for your time. Before we wrap up, we would be grateful if you could answer 2 quick poll questions. If you would like to let us know your thoughts on today's webinar, please feel free to complete our feedback survey in the resource widget or tweet us at BioAnalysisZone. Don't forget to visit Bioanalysis Zone at www.bioanalyseszone.com for more webinars. Thank you again for attending, and goodbye.